Very good evening. We have now quite a few numbers. Good evening to all of you. Thank you very much for being here this evening for this second volume of webinar series on ERA in 2025. So, my name is Vikram, and I'll be moderating the session today. Today, we have Tiffany again here with us. Tiffany will be talking today about exploring the unmatched precision of ERA to further maximize patient results. So, this is, you will understand, you can get it from the topic itself. So, we are not trying to be monotonous here. So we are trying to give you exactly the correct information that you need in your day-to-day practice while dealing with your patients and how ERA is going to help your patients suffering through implantation failures. So before I start the session, uh, I would like to introduce Tiffany. I know many of you in the audience by now must be knowing her, but still there would be few who are new. So allow me a moment, please. Dr. Tiffany has earned her bachelor's degree in animal biosciences from the Pennsylvania State University and PhD in genetics from University of Kent, where her thesis centered on optimizing IVF outcomes by controlling for both embryonic aneuploidy and endometrial receptivity using genetic testing. Tiffany has over 15 years of clinical laboratory experience as a full trained embryologist and is certified by the American Board of Bioanalysts as a technical supervisor in both embryology as well andrology. In addition to clinical laboratory experience, Tiffany coordinated many aneuploidy and single gene cases such as PGTA director at a large IVF clinic in Philadelphia. Tiffany made the move to reproductive genetics in 2016 when she became the PGTL lab manager at Genomics New York in 2017. Her primary focus in the company shifted to endometrial testing when she took on the role of endometrial specialist, concentrating on ERA, EMMA, and ALICE tests. Today, she serves as the Global Endometrium Product Director for iGenomics. Tiffany has published and presented research at conferences such as the Preimplantation Genetic Diagnosis International Society, that's PGDIS, the American Society of Reproductive Medicine, ASRM, the European Society of Human Reproduction and Embryology, that's ESHRE, and the Pacific Coast Reproductive Society. She's an instructor at Eastern Virginia Medical College and is a member of the American Association of Bioanalysts, the College of Reproductive Biologists, and the American Society of Reproductive Medicine. So, this is about Tiffany. Uh, Thank you very much, Tiffany, being here with us again. So I hand over the session to you. But before that, just I request the audience. So whatsoever the questions you have during the presentation of Tiffany, please immediately put it in the chat box, which you can see on top right hand side of your screens. Thank you very much once again. Over to you, Tiffany.
All right, thank you, Vikram, for the introduction. And as Vikram mentioned, today's is volume 2, in which we're exploring the unmatched precision of ERA to further maximize patient results. So I want to start with a brief introduction. So as we know, implantation is a significant rate-limiting step in both human reproduction and IVF. And the equation for success is an optimal embryo plus an optimal endometrium. Now, how do we define an optimal endometrium? Well, there are two main factors to consider here. One is receptivity. Are we synchronizing the embryo transfer accordingly to the window of implantation for the patient, thereby facilitating implantation to be successful? And then there's also microbial health. What is the microbial environment that the embryo will be encountering at time of implantation? Is it ideal where there's low levels of pathogens and high levels of lactobacillus? Or is it pathogenic where we have bad bacteria such as Gardnerella, Streptococcus, and others present, which could inhibit implantation or lead to miscarriage? Now, today we're going to be focusing on the receptivity aspect when it comes to defining an optimal endometrium. So let's look at endometrial receptivity and recurrent implantation failure. We know that RIF affects approximately 10% of couples undergoing IVF. After the third failed cycle, 22 to 50% dropout rates have been reported, with increasing tendency with each consecutive cycle. Patients are desperate to do something different to effectively address and overcome their implantation failures. Essentially, they want a clear solution. Now, 1 in 3 women with RIF have a displaced window of implantation. How can we account for this and facilitate successful implantation in these cases? And this was an answer brought in 2011 with the ERA test. So ERA stands for endometrial receptivity analysis. And it was the world's first molecular assay used to determine each woman's unique personalized embryo transfer timing, therefore synchronizing the embryo transfer with the individualized window of implantation. Now, in 2023, ESHRE came out with their good practice recommendations on RIF. And as far as investigations for RIF, it includes investigations for both female and male factors. However, if we look at the examples provided, we can see that endometrial functioning tests such as receptivity or the ERA test can be considered. Now, ERA is the only endometrial test available that is actually backed by a randomized controlled trial. And if you want to learn more about this particular study, you can go back to volume 1 from our April session. But essentially, this is a RCT that was published in June of 2024 by Barbacadze et al. And here they looked at 3 groups of patients. Our study patients were patients over 35 years old, undergoing oocyte donation with PGTA testing and ERA. Control group 1 was the same age group, so AMA patients, who were also undergoing oocyte donation with PGTA, except they were transferring without using ERA at the standard timing of a FET. And then our second control group, which were younger patients with PGTA and ERA. And what was found was that there was significant improvement in pregnancy rate, implantation rate, and live birth rate when ERA plus PGTA was used versus just PGTA alone, lending to the conclusions of the paper that PET guided by ERA significantly improves pregnancy, implantation, and live birth rates in advanced age patients with challenging reproductive histories. And with no significant difference between advanced age patients, i.e. the study group, and the younger patients, that is control group 2, when using PET guided by ERA, we can see that ERA helps advanced age patients mitigate the effects of endometrial aging. Now, how do we leverage the unmatched precision of ERA to further maximize your patient results? So let's first take a look on how the ERA test was developed. And this is going back to 2011 with the initial paper regarding ERA, where the objective was to create a genomic tool composed of a customized microarray and a bioinformatic predictor for endometrial dating. The second objective here was to define the transcriptomic signature of human endometrial receptivity. Now, looking at the study design, there were 2 separate sets of endometrial biopsy samples taken. The first set was for the ERA gene selection set, and this composed of 20 samples where endometrial biopsies were taken on LH plus 1, LH plus 3, LH plus 5, and LH plus 7. The second set was the ERA predictor training set, um, to define the receptivity gene expression and to define the receptivity gene expression signature. And this was made up of 68 samples taken between cycles day 18 to 12, representing the proliferative stage, LH plus 1 through LH plus 5, representing the pre-receptive phase, and LH plus 7, representing the receptive phase. Now, the results of this paper showed that, well, how did we get the genes in the test? Well, we first analyzed different gene expression profiles in the entire human genome between the receptive and pre-receptive endometrium using raw expression data. Then applying the t-test, we included genes showing an absolute fold chain of greater than 3 and a false discovery rate of less than 0.05. Selected genes were then incorporated into the customized gene expression microarray. And looking at the gene selection, we looked at the intersection between genes statistically significant in groups R versus, so receptive group versus pre-receptive group, and the receptive group versus the proliferative group. We then defined the endometrial receptivity transcriptomic signature by using 134 genes that were specifically expressed during the receptive phase. The predictor gave a specificity of 0.8857 and a sensitivity of 0.99758. So again, I think it's important to start here so we understand how the ERA test was first come to market. Now, just 2 years later, we wanted to show validation of ERA against the gold standard for endometrial dating at the time, which was traditional histology. So here, the objective was to compare accuracy and reproducibility of ERA versus standard histological methods. And again, the study design included two separate sets of endometrial biopsy samples taken. The first set was for the accuracy study, which consisted of 79 samples. We then had subsets from here, one for a training set and one for a dating set. So for the training set, this was for ERA machine learning training and dating, and the dating subset was for comparison between histology and ERA dating. The second main set of endometrial biopsy samples were taken for a reproducibility study and included 7 patients. In this case, the ERA testing repeated in the same patient on the day of their cycle between 29 to 40 months later. So the results show that the concordance of two pathologists against LH surge versus ERA against the LH surge as measured by kappa index. So for pathologist 1, they received a kappa index of 0.618, pathologist 2, 0.685, and ERA 0.922. So to give some visibility of what this means, values between 0.61 and zero point 0.8 mean good concordance, and values of 0.81 to 1 equals very good concordance. So what we can see here is that both of the pathologists performing histological dating had good concordance, whereas the ERA test had very good concordance. As far as reproducibility, if we look to the PCA over here, let me get my pointer, we can see that results were 100% consistent even 29 to 40 months later. So for example, we have patient E, she's both found, I think this is in the proliferative stage, and then patient G, same thing, patient A, you can see that the transcriptomic profiles are very close together, even though several years had passed. So conclusions here was that transcriptomic and bioinformatic technologies applied in ERA confers with a higher accuracy of endometrial dating through the luteal phase and endometrial receptivity, making this molecular diagnostic tool more accurate and robust than the anatomical medicine approach of histological dating. The ERA test is a completely reproducible method for diagnosis of endometrial dating and receptivity status. Now, again, I think it was important to give a little bit of background because I want to show you the evolution of the ERA test. It simply was not just a set it and forget it test. It's something that is continually refined throughout the years. So this next refinement, I think you'll see in your everyday practice, and this was refinement of the transcriptomic signatures. Now, this was a paper published in Fertility and Sterility back in 2017 with the objective to refine the endometrial window of implantation transcriptomic signature by defining new subsignatures associated to live birth and biochemical pregnancy. So again, like our previous studies, we had separate sets of endometrial biopsy samples taken. In this particular study, we had a new training set, which consisted of 450 samples, and these were samples collected at the expected time of receptivity in either an HRT or a natural cycle. We then had an external validation set composed of 321 endometrial samples, and these samples were collected from infertile patients diagnosed with ERA collected at the expected window of implantation in either an HRT or natural cycle. Those patients who were receptive, so this was 228 patients out of the 321 from this cohort, underwent an embryo transfer on the day indicated by ERA, and pregnancy outcome was monitored. So what did the results show? Well, one sub-objective of this study was to build a new training set that was predictive for both HRT and natural cycles. So if you recall the 2011 paper, the ERA test was first designed based upon natural cycles using LH as a reference. The transcriptomic prediction for both was homogeneous and consistent in terms of classification behavior, regardless of the type of cycle. So this is why you're able to use the ERA test in both HRT and natural cycles. Furthermore, unsupervised learning techniques group the former training set, that is the 79 samples from the 2011 paper, according to 6 transcriptomic profiles. So we increased the number of profiles in this paper. So for instance, the former pre-receptive profile, which was LH plus 1 through LH plus 4, was subclassified into early pre-receptive and late pre-receptive. And the former receptive profile, as reference of LH plus 7, was also subclassified into optimal receptivity, indicated by RR in this paper, and late receptive. So again, um, we took we took the initial profiles and classified them into new sub-profiles. And you'll see that more clearly demonstrated on the next slide. So as I mentioned here, you could see we had the initial 3, pre-receptive, post-receptive, pre-receptive, and receptive. And then with this new training, we went from 3 different profiles up to 6 different profiles. Now, what is the relevance of this subclassification? So if we look at the initial test, so this was the one based upon the 2011 paper and the test built upon there, we see the receptive profile, if you see my pointer, is indicated in this black bar. Well, as mentioned, we then divided this into separate sub classification of transcriptomic profiles. And essentially what we did was we defined an even more optimal receptive phase. Now, I think this is very important because, again, it's a clear demonstration on how the ERA test has been continually refined to give the most precise results and recommendation over the years. So looking at this optimal receptivity signified RR, we can see higher ongoing pregnancy rates, higher implantation rates in this group, higher clinical pregnancy rates, and much lower biochemical pregnancy rates. And this becomes very obvious when we look at the late receptive phase. So the late receptive phase is a phase that we have already incorporated in our ERA test today. In fact, it's been incorporated for over 8 years. And we look at the results associated with this late receptive profile, we see a 50% biochemical pregnancy rate. Now, when we compare that to optimal receptivity, there's only a 6.6% biochemical pregnancy rate. So this is our first clue that the late receptive profile has a strong association or a strong risk of biochemical pregnancy. And how I explain this is that when we transfer during the late receptive phase, we can see that we're starting to be toward the end of receptivity. So maybe something is happening there as far as implantation. We're just not getting a good implantation. So we have that initial positive pregnancy test and then a loss from there. So this is really important because, as I mentioned, with the initial version of the ERA test, um, these subprofiles would have been classified as receptive. And after this refinement, we saw, yes, there is indeed a separate transcriptomic profile that we must pay attention to, and that's clinically relevant for our patients. So the conclusions here was that major difference between window of implantation transcriptomic stratification were in the ongoing pregnancy rate and biochemical pregnancy rates. The ongoing pregnancy rate ranged from 76.9% and 80% in the late pre-receptive and um, optimal receptivity signatures, respectively, versus only 33% in the late receptive profile. The biochemical pregnancy rate was also significantly lower in the late pre-receptive and optimal receptivity being 7.7 and 6.6% respectively. However, in the late receptive profile, there was a 50%, that's 50, biochemical pregnancy rate. These findings have permitted the improvement of the predictive accuracy of the ERA gene signatures, making it possible to detect the best transcriptome associated with successful pregnancy. So again, what is the ideal gene expression that's going to be associated with the most optimal outcome for patients? And that was what was further defined here in this paper. And then lastly, a new subsignature was discovered that may help prevent biochemical pregnancy of endometrial origin. So again, I do want to take a second because I think that this refinement here can be well seen in your own day-to-day practice. So again, what is the significance of having a late receptive profile? Well, from the previous slides, the late receptive phase consists of 22 dysregulated genes and associated with significantly higher rate of biochemical pregnancy loss. When a late receptive result is obtained, the recommendation is to transfer 12 hours earlier than when the biopsy was performed. So for instance, if you took the ERA biopsy at 120 hours in an HRT cycle, and a late receptive result was obtained, the PET recommendation will be to transfer 12 hours earlier at 108 hours. So just this minor shift of 12 hours is going to get your patient back into that optimal receptivity range. ERA is the only endometrial receptivity test that provides results at 12-hour increments. So if you look at other endometrial receptivity tests that are out there, a lot of them will say just transfer P plus 5 or P plus 6. Results are typically given in 24-hour increments and usually on a day-to-day basis versus hour to hour. So hence, when we're using the 12-hour increments, we're precisely identifying and counting for the personalized window of implantation of that patient and potentially avoid adverse outcomes such as biochemical pregnancy loss. Furthermore, transition phase profiles like the late receptive result can be particularly important in patients with a narrow window of implantation. And how we define that here is less than 24 hours in duration. So we expect the window of implantation to be about a day, maybe a day and a half long. However, we've seen patients with window of implantations that are less than 24 hours in duration. In fact, we've seen patients where we've seen less than 12 hours of duration. So for these cases, it's very, very important that we're transferring at the optimal time for those individuals because they don't have additional time to spare for the embryo to just float around and wait for receptivity. We need to target when that patient is going to be receptive, and we need to plan accordingly in order to ensure proper synchronization to increase the likelihood of implantation and decrease the likelihood of adverse outcomes such as biochemical pregnancy loss. So with profiles like these, like the late receptive result, where we're just moving at 12-hour increments, we can give the most precise timing to patients and thus have the most optimal outcomes for that individual. Now, what other improvements have been made over the years? Well, in 2017, we moved from a customized microarray where we analyzed expression of 238 genes to next generation sequencing, where we now analyze the expression of 248 genes. And the number of genes incorporated into ER tests is also very important. We know that endometrial receptivity itself is a very complex matter. So we would assume that we need to understand more than just a handful of genes what's going on here. There's lots of things at play. So we found that having several hundred genes included in the ERA test is going to give us a best picture or best view of the endometrial receptivity or window of implantation in patient analyses. Now, next generation sequencing offers a high throughput sequencing, allowing for shorter turnaround times. And furthermore, unlike a customized microarray, changes to the gene panel can be done quite quickly without having to redesign the customized microarray each time. So for example, we add an additional 10 genes with the NGS version versus the initial microarray that had 238 genes. Now, this was great that we switched to NGS. However, probably one of the biggest advantages of when moving to NGS was the retraining of the ERA algorithm or the ERA predictor. So this is the computational predictor that actually gives us the clinical directive associated with ERA, i.e., when to transfer. Do we need 12 hours less progesterone? Do we need 24 hours more progesterone? Do we need 48 hours more progesterone? And initially, a cohort of endometrial samples along the menstrual cycle were used to train the predictor for endometrial dating and to define the transcriptomic signature. So, i.e., this was the 2011 paper that we discussed a few slides ago. However, when we introduced the NGS version, the ERA predictor was later updated to include controls from patient cases with a receptive ERA profile and when transferred according to their personalized window of implantation had successful pregnancy. So essentially, version 1 was just based upon gene expression profile only. So when we performed the ERA test, we looked at the controls within the test and we compared the transcriptomic profile of the sample to the controls and essentially where did they fall? Did they fall into the receptive range, the pre-receptive range, the post-receptive range, etc. But when we introduced the next generation sequencing and the updated algorithm, um, not only were those receptive profiles show a receptive transcriptomic profile, but those patients who transferred according to their ERA results also had successful pregnancies. So essentially, when you get the ERA results today and a receptive profile is obtained, those results are based also off of clinical outcome, not just simply transcriptomic studies. So this is very important to keep in mind, and this certainly leads to the accuracy and precision of the ERA test. Now, another update or evolution of the ERA test over the years was that results were reported in hours and no longer days. So this switch was in 2017. If you've been performing the ERA test, prior to 2017, you probably remember the earlier version. We used to give the ERA results and PET recommendations in days. So if a patient, say, had a biopsy on P+5 in an HRT cycle and she was receptive, we would say transfer on P+5. If she was pre-receptive by 1 day, At that time, we actually used to recommend another biopsy. Now we don't. We give a direct recommendation for transfer, but we would say, okay, biopsy 24 hours later on P plus 6. Well, as the ERA test has been refined over the years, standard reporting is now in hours, permitting a more precise timing. Again, this is especially important in those cases of where patients might have a late receptive result and we want to avoid biochemical pregnancy, or in patients that possess a very narrow window of implantation of less than 24 hours. So we really have to hone in on when that patient has optimal receptivity. And to do this is following the report in number of hours. So for this reason, this is why we ask you to provide us with first intake date and time of progesterone in HRT cycle, or maybe HCG triggered a natural cycle, and the exact date and time of the biopsy. We can calculate the total number of hours the patient has had exposure, for example, an HRT cycle to progesterone. So maybe the patient had, in the previous reporting, maybe it would show that her biopsy was on P plus 5, but being that P plus 5 is a 24-hour increment, essentially, there might be a huge difference between performing the biopsy at 108 hours versus 120 hours, which again, both of these represent a P plus 5 timing. So having these exact timings, we can, again, precisely understand when the patient has optimal receptivity and give a precise timing recommendation for transfer. So in the case here to the right, this patient had 121 hours of progesterone exposure. And again, we calculated that based upon progesterone start time and biopsy time. And we see she's receptive. So we want to perform that transfer at 121 hours plus or minus 3 hours. We do have a little bit of wiggle room, but we want to be within that time frame. Whereas if we look at the previous results prior to 2017, we would just simply say the patient had biopsy on HRT P plus 6, transfer at P plus 6, which could be a game changer for patients if they do have a narrow window or are prone to late receptive transcriptomic profile. Now, we're continuing learning from patient cases worldwide. So powered by AI, the ERA algorithm has continually been refined as more endometrial samples are analyzed. Unmatched in the number of cases, over 200,000 women worldwide have undergone the ERA test, lending to a superior accuracy. And I think this is very important to keep in mind because, as anything, I think we all have AI with some form in our life now. The more we feed it, the better it gets. And that's essentially the key message here. The more we continue to learn, the more samples we have, the more experience we have, the better the test gets. What about further clinical and scientific support for ERA? So ERA has been extensively studied since it was first launched in 2011, with over 50 clinical trials supporting its role in personalized embryo transfer. We've certainly learned from the clinical and scientific support that we've gotten with ERA. So much like the number of patients who have undergone the ERA test, we take this into consideration and we continually refine the test with more data that comes out. And then finally, continually improving with cases reviewed by our endometrial experts. So proper protocol compliance is key to best patient outcomes for ERA. Our unparalleled endometrial team reviews patient cycles and results to ensure accuracy, and accounting for factors such as proper endogenous progesterone control and cycle protocol replication provides the most precise results. And I know sometimes we get complaints about maybe the overwhelming amount of information that we ask for when it comes to our ERA test, but this is very important, and this is a reflection of the review that our endometrial experts do on as we receive cases for ERA. We want to ensure that the sample was taken properly. We want to ensure the accuracy of the information that we're provided because we know that this information and taking this information into the analysis of the results is imperative to give the best recommendation for each patient. So if you want to refer back to our one of our earlier webinar series, this was actually a webinar series from 2024 where we looked at proper protocol compliance. I have an example of what happens if endogenous progesterone isn't properly controlled for. And essentially what could happen is that maybe we're transferring outside the window of implantation. So the patient isn't getting the benefit of receptivity testing in those cases at all. And in fact, when the protocol is not followed, it's a bit wasteful for the patient's time and expenses. So it's very important that we follow proper protocol because we do take the information you provide us. As I said, we do checks to make sure that it all adds up. Sometimes if we see a missing endogenous progesterone, we'll ask for this information, or if endogenous progesterone is taken not within the 24 hours prior to first intake of exogenous progesterone, we'll follow up asking, do you have a more updated endogenous progesterone information? Or if we see that perhaps the cycle is taken in a simulated cycle, which is not supported by the ERA test. We'll come back and we'll say, okay, this may affect the reproducibility of the results and create a bias in the results that we obtain. So we recommend canceling and taking it in a properly validated cycle type, which would be HRT or natural. So we do a thorough review of all patient samples received, making sure that we can give the most accurate answer to those patients. And as I mentioned, protocol compliance is key to optimal ERA outcomes. We have a newly updated ERA Insight Hub that has resources for both clinicians and patients. So this was just revamped in the last month. We have ERA in Focus, which right now we're featuring the RCT with key findings, including an interview with the first author. Additionally, ERA has been featured in new Rescripted articles, which Rescripted is a great resource for patients to understand not only IVF, but women's health. We have tips from our scientific advisors, along with tools that you can use on your day-to-day to make sure you're optimizing the ERA's results as much as possible. This includes our PET calculator, ensuring that you're performing transfer at the correct time. And then our ERA clinical tool, which makes sure that you're replicating the cycle appropriately between the ERA mock cycle and the ERA transfer cycle. Our ERA in practice features webinars of current webinars, so you can register for our third webinar that we're having next month on June 21st, along with previous webinars, which includes that protocol compliance webinar I referenced in a few slides ago. We also have ERA publication list, along with a new paper that was just published 2 weeks ago looking at PGTA plus ERA together and finding significantly better outcomes for patients when we're doing a dual approach to IVF. And then lastly, this is a great resource for patients. We offer endometrio pre and post testing webinars with our endometrial nurse that's based in the US. And patients have the opportunity to have their questions answered and learn more about ERA, Emma, and Alice. So this is a complimentary webinar available to patients. Now I'm going to round this out with conclusions from this presentation, then we'll open up for question and answers. So this is obvious, but I think important to emphasize. Successful implantation is a rate-limiting step to successful pregnancy in both human reproduction and IVF, and largely hinges on an optimal embryo and endometrium. RIF affects 10% of the population, and patients affected by RIF are prone to drop out of IVF treatment and are eager to address the cause of implantation failure. ESHRE good practice recommendations indicate that endometrial function testing, such as ERA, can be considered in cases of RIF. The ERA test was first commercially available test addressing RIF of endometrial origin and the only endometrial receptivity test supported by an RCT. Since the introduction of ERA in 2011, the test has continually been refined to provide the most optimal outcomes for patients. The ERA test is the most widely used endometrial receptivity test worldwide, which greatly lends to its unmatched precision. ERA is unmatched in the number of scientific publications that support its use, with over 50 papers published. Robust research surrounding ERA also provides a means to the test refinement. Careful review of cases by our endometrial experts ensure accuracy and precision of patient results. Protocol compliance remains key in ensuring the most optimal patient result outcomes. And then the final note I want to make, so as as I mentioned, ERA was the first commercially available test of its kind. Since then, competitors have come out with new tests, and a lot of these competing endometrial receptivity tests are validated against the ERA test, so ERA being the gold standard. However, it's important to ask or consider what version of the ERA test was used in their validation. Was it version 1 from 2011? If so, they're lacking key refinements that have been made over the past 14 years, making their tests severely outdated. So with that, I want to thank you for your time today and welcome Vikram back, and we can start the Q&A.
Thank you very much, Tiffany, for your insights. I'll go to we have few questions here. I'll start with the first question. That is, while estradiol can be given for max 17 days, is there a limit of minimum days of estradiol exposure?
So we recommend no more than 17 days of estradiol before starting progesterone. And the reason for this is that extended estradiol administration is associated with endometrial bleeding. And in the case of ERA, we found a link of endometrial bleeding with non-informative profiles. So this is when we get a profile that does not match any of the controls within our test. And then the patient has to come back for a new ERA cycle and rebiopsy to get an informative result. Now, when we do receive a non-informative result, the good news in over 95% of rebiopsies, we're able to get an informative result for the patient. So that means that something happened in that particular cycle that led to this non-informative result. And as I mentioned, endometrial bleeding can be one cause of that. So, in order to avoid that, we say no more than 17 days of estradiol priming before uh, before progesterone start. Now, as far as a minimum, um, we see maybe 7 days of estradiol priming is sufficient, as long as you're building an endometrium that is normal for that patient and that you would feel comfortable transferring in. It's fine.
Does the doctor understand, by virtue of your reply, that if the endometrial thickness is 7 mm at day 8 or day 9, the doctor can proceed with the P4 administration?
Yeah, I would, if that's normal for the patient, so if the patient has a normal endometrial lining defined as 6 millimeters to 12 millimeters from cycle to cycle, and that's endometrial thickness that you would feel comfortable transferring in, then 7 days is sufficient to then start progesterone.
Thanks, Tiffany. Tiffany, as I think this question pertains to your one of the last slides, wherein you talked about pre 2017 and post 2018 and change of algorithm. So the question is, if error test is valid, as how long is error report valid for, and would the change in algorithm have bearing on report of receptivity?
Um, so the ERA test we say is good for about 2 years. So we've seen that even though it's been reproducible up to 40 months, um, we recommend repeating after 2 years. And the reason for this is because if a patient has any change themselves, for example, a change in BMI, we know that BMI is greater than 30. These patients have a significant displacement of window of implantation. So if you have a patient who maybe had a BMI of 20 in their first ERA and now has a BMI of 25, we can expect that maybe the transcriptomic signature has been affected. Furthermore, maybe the patient has had some alteration to their uterine cavity that we need to consider that could affect the transcriptomic signature. But if you're within that 2 year range and there hasn't been significant change to the patient herself, and you're ensuring that you're replicating the protocol exactly, the results are expected to be reproducible. And I'm sorry, Vikram, the second part of the question I forget.
No worries. Yeah. Uh, if there is any change in the algorithm, would it
Ah, ah,
have any bearing on the report of receptivity?
I see. So when there's major changes, so like for example, adding the late receptive result, if the result was prior to 2017, that would have been reported as receptive. And now post 2017, it is late receptive. So there can be some changes in the recommendation. Now that was a major, I would consider that a more major update than some of the other refinements we had, but we do make sure that we announce to our clients these updates when they come through. So in 2017, you had an update that we switched to NGS. Also in 2017, I didn't include it in this presentation, but we also had an update where if you previously remember, when we had a pre-receptive 1 day profile, we used to say, okay, biopsy with 24 hours more progesterone. Well, in November 2017, after further study, we were able to have a higher confidence saying, okay, instead of biopsying 24 hours later, you can go ahead and transfer 24 hours later. So we were saving the patient an additional need of a cycle, and they could proceed with transfer in the following month. So these changes are communicated with our clients, but I, if you do have samples prior to 2000, or if you do have results prior to 2017, I would recommend repeating them anyway because it's been now 8 years. So potentially the patient has had some changes herself.
Thank you, Tiffany. Tiffany, this is another question seeking validity of error results, which you have already addressed. The second part of this question is, do we have any data after downregulation with Zoladex in severe adenomyosis patients?
Um, I don't know. I'm guessing that is a medication. I would have to look
Yeah,
into
it's,
that.
it's basically, I think, either GnRH agonist or GnRH antagonist. Either of the two, I don't know exactly what Zoladex is, but it is a GnRH, drug. So,
Okay.
do we recommend down-regulating in error cycles with GnRH?
No, so I mean the optimal ERA cycle would just be with estrogen and progesterone only. When we add additional medication, we take the risk of adding artifactual results. Now, it's hard to test the effects of every single medication on ERA because, for instance, I'm based in the U.S. and the medications we have here may not be the same medications you use in India, or the same medication used in India may not be used in Europe, and et cetera, et cetera. So it would be impossible to assess the effects of every single medication on the ERA results. However, if there are medications that you absolutely must use, our recommendation is to replicate that protocol exactly between the ERA mock cycle and the ERA transfer cycle to avoid any potential artifact of those medications. But ideally, um, you want to keep the cycles as simple as possible, and really estrogen and progesterone would be best, but we understand that sometimes there's things that are unavoidable and additional medications must be used. Now, we don't want to use any stimulation medications. We don't want to have any ovarian stimulation, because this can add an artifact itself. So this can lead to premature progesterone exposure, amongst other things that would give us inaccurate results in some cases.
I hope doctor's question is addressed. Tiffany, this is about if a patient is taking steroids in Aracycle, uh, should she continue taking steroids in the PET cycle? Would it have any adverse effect if there is no need of taking steroids while performing the PET?
Yeah, so again, we would recommend replicating the mock cycle and transfer cycle, and this would include the steroid use. So if the patient is under steroids for the mock cycle, then you want to use the same protocol for the transfer cycle. Ideally, if it's just for if she's on steroids, maybe for something outside of her IVF cycle, perhaps it would be ideal to have her finish her steroids for that and then do the mock cycle after with only estrogen and progesterone as an HRT. That way it could be replicated exactly in the transfer cycle. But again, we do recommend replicating everything exactly just so we can omit a potential artifact to the results.
Okay, thank you, Tiffany. Uh, well, next question is what is the incidence of displaced error in endometrial dysbiosis diagnosed with Emma?
That is a great question. I don't have that information readily available. There has been some studies indicating that a dysbiosis or an infection in the endometrium can lead to a displacement of the window of implantation. So this is something that we're studying along with other clinics. I believe there's a recent paper from Japan, if I'm not mistaken, on this. But it's something that we don't entirely know the effects of an altered endometrial biome on the window of implantation. So uh, that's a good question. I'm going to ask our laboratory team to see if they can give me statistics on EMA abnormal and receptivity. But
Tiffany,
in
I'll share the email ID of the doctor who has put this question. Please revert to him as early as you can. I'll share his email ID today. I know maybe Monday you can revert to him if it's okay.
That would be perfect, yeah, because I think that's really interesting. We see in general the displacement is about 1 in 3 patients with RIF, but that's without considering or looking further at the endometrial microbiome of those patients.
Thank you, Tiffany. There is a doctor in the audience. A doctor is inquiring about the cost of error. Doctor, be assured that immediately once this we finish with the session, you will get a call and we'll share the cost of error with you. Thank you very much. Certainly on priority. Well, Tiffany, this is the last question. It's if we have day 4 embryos and error receptivity is defined as p plus 5, then how do we handle this?
Yeah, this is a very good question, um, because there is some confusion that we see sometimes about, okay, if we have day 3 embryos or day 4 embryos, how do we perform the ERA test? Well, you're always going to want to perform the ERA test as if you're transferring a blastocyst, because this is the stage of embryo that implants. So if you're doing an HRT cycle, you're always going to take the first biopsy on P plus 5 or 120 hours of progesterone exposure. Now, in order to adjust for the stage of embryo, you would just subtract the appropriate amount of time from your results. So let's say that you perform an HRT cycle, perform biopsy at 120 hours of progesterone exposure, and the patient is receptive. The report's going to specifically say transfer a blastocyst at 120 hours of progesterone. But in your scenario, you're transferring a day 4 embryo. What you'll simply do is just subtract 24 hours from the recommended time of blastocyst transfer. So in the example we're working with, blastocyst transfers at 120 hours minus 24 for day 4, your transfer would be at 96 hours of progesterone exposure. The same thing you would do if you were transferring day 3 embryos, but instead of subtracting 24 hours, you would subtract 48 hours. Um, and that would be, let me see if I can work out in my head, 100, or it would be 72 hours of
72.
progesterone exposure for day 3.
Yeah. Thank you, Tiffany. Tiffany, one more question is there. Will change in duration of estrogen priming prior to progesterone exposure alter the outcome even if progesterone supplementation was well replicated?
Yeah, so you have a little bit more freedom with the estradiol priming of the cycle. So for sure, progesterone protocol needs to be replicated exactly and adjusted for the results if necessary. For example, if you need to add additional 24 hours of progesterone. But when it comes to estradiol, you can add some days, take away some days if necessary. The point here is that we want an endometrium that is similar from cycle to cycle. So let's say in the ERA mock cycle, you did 10 days of estradiol priming before starting progesterone, and you had a lining of 8 millimeters. But in the transfer cycle, you're on 10 days of estradiol and you're only at 5.8 millimeter lining. You can give several more days of estradiol priming in order to get between 6 to 12 millimeters within that normal range of endometrial thickness. So that's the more important part when it comes to estradiol priming. Is that estradiol priming protocol giving you a comparable endometrium from cycle to cycle, and you can play with a number of days in order to obtain a endometrium that is similar from the mock cycle to the transfer cycle. But once you start progesterone, the protocol needs to be exactly the same and only adjusted if according to the ERI results.
Thank you very much, Tiffany. Thank you very much to the audience for joining us today. Truly, we appreciate your time and participation, your questions. Thank you very much. And I would like to thank Tiffany also for taking her time out and sharing her valuable insights with the esteemed audience. Thanks to the people like NG, who is working behind the scenes for making this possible each and every time. Thanks, NG. And for the audience, I assure you, you will be receiving the recording of this session very soon on your email IDs. And we hope to see you at the volume 3 of ERA webinar, and you will get the notification prior to that. Thanks again. Thank you, everybody. Take care and stay connected. God bless. Bye bye. Thank you, Tiffany.
Thank you, Vikram.
Bye.